Now, the first type of operand system let's discuss is the inducible operand system. Inducible means something which will be stimulated to become operational or functional or switch on. So that means these genes are generally, or these operand or this unit of which functions together. That's why it is called operon, because it functions together under same operator. That's why it is called as operon. So inducible operon is switched off generally, but if the inducer comes into the environment, that's why it is in the last page we read from outside. So if the inducer comes into the environment, so a stimulus is received, and now the switched off gene will be turned into on. So switching on of a gene or switching on of a number of structural genes, because in this topic we'll be learning about 3 genes functioning together. So the switching on of the operon in the presence of inducer is called as inducible operand system. Now let's understand some of the genes which are important constituent of this system. So the inducible operon example of the NCERT is the lac operon of E. coli, and lac stands for lactose. Remember, lactose is a disaccharide, so it has to be digested, or we should say it has to undergo catabolism to form one molecule of glucose and one molecule of galactose. That means inducible system or lac operon is generally come into catabolic pathway. So lac operon is an example of catabolic pathway. So the inducible system consists of 4 types of genes, and then there are some chemicals also involved. So the first gene is the structural gene, which is also called as the cistron. Now what is the structural gene here? In this picture you can clearly see it's exactly copied from the NCERT. You can see there are 3 structural genes I have mentioned here. So the sequence has to be same because it is as such taken up from the NCERT. The genes are Z, Y and A genes. Now this O stands for operator, P stands for promoter, I stands for regulator gene, and P stands for the promoter of this regulatory gene. So this is the promoter of the regulatory gene, this is the promoter of the operator, and then these are the 3 structural genes. This is the example of lactose operon or lac operon. So structural gene, now how do you define a structural gene? Structural genes are the segment of DNA which carry the codes for synthesis or synthesizing the proteins. That means these genes determine the primary structure of a polypeptide chain by having the coding sequence of amino acid in them. We all know DNA codes for amino acid. DNA will have codon. Codons will give rise to mRNA codon, and mRNA codon will guide the sequence of the or primary structure of the protein. We have already covered this in the last class. So structural gene synthesizes the mRNA, which in turn controls the polypeptide or enzyme formation over the ribosome. Now for example, the lac operon has in the E. coli, Escherichia coli, contain 3 structural genes, that is Z, Y and A. So there are 3 genes involved. Now you can see here I have already mentioned Z, Y and A. Now all these 3 genes code for different enzymes. That means they will be causing the formation of different enzymes which I have mentioned here. The first gene is gene Z. Now this gene codes for the enzyme called as beta galactosidase enzyme, and its function is to break down the lactose into one molecule of glucose and one molecule of galactose. Now the second gene is the Y gene. The Y gene codes for the enzyme called as the Permease enzyme. Now Permease enzyme, permease means permeability. This enzyme is a membrane bound carrier protein which help in the transfer of the metabolites or molecules which are similar to inducer will come inside with the help of the permease enzyme. We'll be learning about this in detail here. Then the third gene is the A gene, which synthesizes or which codes for the enzyme called as trans-acetylase enzyme. Now its function is to transfer. If you remember in 11th class, you have done transferase enzyme category. so it belongs to the same category. Transfer of acetyl group from coenzyme, transfer of the acetyl group from the acetyl coenzyme A and transferring it to the galactose is called as the trans-acetylase enzyme. So it is supposed to transfer the acetyl group. To be more precise, this gene or this enzyme does not play any role in this metabolism, but because they are present together, so they always function also together. That's why NCERT also explain these three genes together. Now the next thing is